I would like to welcome you all to the CCG webinar workshop series. So thank you all for joining us. Today's presenter is Mike Drummond, Principal Scientist at CCG. And just to introduce myself, my name is Ansen Tan, Application Scientist at CCG, and I'll be moderating today's webinar. So without further delay, I'm delighted to introduce to you Mike Drummond, who will present today modeling ternary complexes with molecular glues, making sense of a sticky situation.
All right, thank you very much, Antien. Today we're going to be talking about molecular glues and how we can model complexes containing them within Mo. I'll start off by giving a quick introduction to what molecular glues are, and then I'll look at some of our initial efforts at CCG at modeling these complexes that contain molecular glues. And then we'll move into a slightly different tactic in modeling these complexes, considering them as linkerless bifunctionals, and I'll explain what I mean by that. And then we'll conclude, and then probably have some time for a little bit of Q&A. So let's go ahead and start by talking about what molecular glues are. The term molecular glue first appeared in the literature in 1992, and it was when Schreiber and co-workers were looking at these two proteins, the FRAP and FKBP12, that were known experimentally to associate with one another. However, when Schreiber et al. Took a little closer look, they could not find any evidence of a direct contact between these two. It was only when a small molecule, rapamycin, was introduced that they found that rapamycin first interacted with FKBP12, and then once that formed, the complexes came together to form a three-component or ternary complex. And so they coined the term molecular glue, meaning a small molecule that facilitates a protein-protein binding interaction that otherwise wouldn't occur without the glue present. So you really are just gluing these two proteins together. Today, we know that glues can stabilize a protein-protein interaction, but the reason there's so much interest in this topic, particularly in therapeutics, is through their application of what's known as targeted protein degradation. And I'll explain what that is on the next slide. Today, targeted protein degradation, or TPD, is dominated by heterobifunctional degraders, such as this PROTAC, Arvinas ERV110, shown down here at the bottom, as well as molecular glues. And over here on the left, I've shown molecular glues that are either on the market or currently in clinical trials. And if you compare this set of molecules to this one over here on the right, you can see why we're interested in glues. They tend to be smaller and less flexible than heterobifunctional degraders, such as this one over here. They're generally within standard rule of 5 parameters, although if I show the molecular weights for these glues, you can see that sometimes we creep over 500 grams per mole, but still quite a bit less than even some of the smaller degraders, bifunctional degraders shown over here. I will say something important to understand about glues is we understand them to be monovalent. And what I mean by that is that the entire molecular glue simultaneously interacts with both proteins. And that's in contrast to a bifunctional degrader, such as over here, where you have one end that clearly interacts with one protein, and then the other end is its primary responsibility is interacting with the second protein. With glues, the entire molecule interacts with both proteins at the same time. And so because of that, rational design of glues has proven elusive. Now, you probably noticed, if you take a look at this entity over here on the degrader, you probably noticed that there's something quite similar on many of these molecular glues. On this bifunctional degrader, this is the part that binds to an enzyme known as E3 ligase, and it's the same moiety or very similar moiety over here on the glues. And the reason we have that commonality is because in targeted protein degradation space, particularly with glues, almost all the time currently, at least that's been disclosed, we are working with an E3 ligase known as cereblon. So what does this all mean? What is cereblon? What's an E3 ligase? Cereblon, shown here in green, is just one part of a much larger assembly known as the CRL4 cereblon E3 ligase assembly. And this whole mess of assembled proteins, its entire job endogenously is to bind to a protein of interest, and then ubiquitin gets transferred over to that protein of interest, that POI. And then once you have enough ubiquitins transferred over to that POI, that is the native signal for that protein of interest to be sent off to the proteasome and degraded back into its peptides. Now, what we're doing with a molecular glue is we're hijacking this system so that we are not restricted to the naturally occurring protein of interest. Instead, it's going to be whatever target protein we hope to affect, and in particular, whatever target protein we would like to degrade. And you can understand why we're interested in degrading these proteins rather than just inhibiting them, because if our POI has a deleterious effect, it would have to actually be resynthesized in order to show that effect. Whereas with an inhibitor, as soon as that inhibitor comes out of the pocket, the protein can misbehave as it will. And so obviously there's some advantages with targeted protein degradation. I will say that TPD operates on, you don't have to have an enzyme protein. It can operate on a scaffolding protein. It's also catalytic. Multiple copies of a POI can be degraded, and it's quite selective as well. And this holds true both for molecular glues and for bifunctional degraders as well. Now, if we talk about the main component that I'm going to be interested in modeling within Mo is that ternary complex, that 3 component. And to make our life simpler, we're really going to talk about those 3 components would be the glue, the protein of interest, and then rather than this whole mess of proteins, really just kind of the substrate receptor at the end, such as cereblon right here. And so before those 3 components come together to form a complex, a binary complex has to form first. This is true both in reality as well as if we're looking to simulate the situation. Very often we know the order of events that happens. For example, with cereblon, we know that the glue first binds to cereblon and then the POI comes in. However, we maybe shouldn't make so many assumptions if we're looking at novel systems involving enzymes or proteins beyond cereblon. If we're looking to explore and develop new glues for new degradation or new stabilization, things like that, really the fewer assumptions, the better. And so what that means is we can conceptually just enumerate 3 different ways that we can look to model ternary complexes. One might be that E3 ligase and or whatever protein and the protein of interest can come together first, and then the glue comes in as a second step. Or perhaps, as with cereblon, the glue first interacts with the E3 ligase, and then the POI comes in. Or maybe for the sake of completeness, we can model it with the glue first binding to the protein of interest, and then that E3 ligase comes in as a second step. Now, this first one I enumerated might seem a bit odd because I told you in one of my first slides that glue facilitates a protein-protein interaction that wouldn't otherwise occur. However, there has been some recent work, and here's a couple references down here, where they actually find a baseline level of weak but certainly measurable protein-protein interaction between your 2 proteins, even without the glue present. And so it's thought maybe the glue is enhancing some sort of native PPI as opposed to creating it from scratch. And so because of that, maybe this isn't such a silly thing to evaluate. And on top of this, as we were looking to model these, we didn't want to close any avenues of discovery without investigating them. So we did try all 3 of these order of events as we're looking to model these. Of course, the way we find out what's the best way to actually form these is we wanted to do this empirically. And what I mean by that is we put together a data set. We gathered it from a couple of references shown down here of known structures, almost always a crystal structure, some cryo-EM structures of ternary complexes containing glues. And so here's the entire list. We'll have a book chapter coming out pretty soon that goes into more details on this. But here's their PDB codes. And a quick look will tell you that many of these, 8 out of the 32, do contain cereblon, so they're directly relevant to targeted protein degradation. Another 7 of these structures contain a different E3 ligase known as decaf15. And so again, relevant, about half the data set is relevant to targeted protein degradation. Another 11 were from recent work from AstraZeneca, where they looked at stabilizing interaction between a protein and a peptide, and then the remainder of our data set looked at endogenous glues. These are natural products like auxin and rapamycin and things like that. And so in order to really, we're just going to see if we can reproduce these, predict the structure of these, compare them to the known answer, and see how well we did with the various approaches that we're developing. So our first approach this is our first attempt at doing these, so we called it approach 1. And it really was just starting with that first method of assembly I enumerated before. What if we take the E3 ligase, or it doesn't have to be an E3 ligase, just one protein, and the second protein, we bring them together, and then the molecular glue is added as a second step. And so the computational tool that one uses to put two proteins together is known as protein-protein docking. And so we use Mo's engine to do protein-protein docking, and by default, you get 100 total poses, total possible hypotheses for how, for example, the magenta protein can interact with the green protein here. And here I've just shown all 100 possible solutions. Now, it's also possible as you do the protein-protein docking, both just using Mo's protein-protein docking or in our molecular glue tool, which I'll show you in just a few slides, you can optionally add constraints. If you happen to know or suspect even that just one portion of one or both of these proteins interacts with the second protein, then you can add that as a constraint. So for example, if I think that this area of the green protein is really where the magenta protein is going to interact, I can enforce that with a constraint, and that would therefore remove some of these outlying poses where it's not interacting with that central portion of the green protein. It is optional, however, but if you do have that information, we do suggest that you give it to the simulation. One way or the other, with or without these constraints, as you do the protein-protein docking, by default, you get 100 total poses. What we then do with approach 1 is for each and every one of those poses, so we'll take just a single one here, we then use Mo's site finder tool, which will find pockets. And what we specifically are looking for are pockets that are within 4.5 angstroms of both proteins. So in general, these are sandwiched, buried in between the two proteins, or sometimes near enough, as I've shown here, that it's not exactly in between the two proteins, but it's quite close to both proteins. Once we have that can serve as a docking site, and we simply do small molecule docking to put a putative molecular glue into that identified site. We take the single best scoring pose using Mo's default scoring function, and that's your ternary complex. That is the three components: protein 1, protein 2, and the molecular glue. And we just repeat this for the 100 poses of the protein-protein dock poses, and we give you a library of 100 possible answers. Now, before I talk about the results here, I do want to define metrics for success. I said we're going to compare these modeled results to the known experimental ternary complex structures, and so I have to define metrics to define whether we're successful or not. And it's really the same metric, we just applied in two different ways. We're going to compare it to the crystal structure, and we're going to define a successful hit, a successful prediction, as for the protein, the alpha carbons of that docked protein have to be within 10 angstrom RMSD of the known crystal structure. So to illustrate that here, I'm for a slightly different system or slightly different perspective as well. I'm showing all 100 predicted poses of that protein. The red ones are incorrect, really hard to see, but buried in there somewhere is the crystallographic pose in green. So if I hide those ones that are more than 10 angstrom RMSD, the yellow ones are the ones that are within 10 angstrom RMSD. And you can see they're in the right ballpark, I might say colloquially. So we got it, kind of close, or actually fairly close. If your protein is larger, it actually looks quite reasonable just from eyeballing it. So that's our first metric of success. We use the same thing for the glue. So same sort of thing, the green position and space filling, that's our crystallographic answer. The red ones are just incorrect, more than 10 angstroms, and the yellow ones are again close enough for us to consider it a hit. Now, let's take a look at those hits. I told you by default, we get 100 poses. It doesn't seem right to me to just give you 100 answers and say, it's probably one of those, good luck. Whether it's pose 1, pose 20, pose 99, that's up to you to figure out. No, what I'd really like to do is be able to give you a score, so you can have some confidence that if you're sorting this output database of poses, that it's going to be the ones at the top and not the ones at the bottom. So as we do this ternary complex modeling, you actually, with this approach 1, you actually get two natural scores. The protein-protein docking phase gives you a score, as does the molecular glue, that small molecule docking phase. And so we can look at both of those scores to see if one or the other does a better job of giving us accurate results. So here for that 32-member test set that I talked about, we're looking at 4 different scenarios, if you will, where we either added constraints on neither protein or both proteins, or maybe we just added a constraint on the larger protein or the smaller protein. And we added this constraint based on the fact that we know the answer from the crystal structure. So it's meant to simulate as you're advancing in a project, maybe you get a little bit more information about your system. So if we look at the 32 different systems, each one gives you 100 poses. What if we look at just a single prediction for each of those 32 that has the best protein-protein docking score? And this line here is saying out of those 32 answers with the best protein-protein docking scores, 32 predictions, how many of them have the protein about right? And you can see it's generally about 2/3 of the time. We do a little bit better job if we constrain both proteins, but not a whole lot of effect there. But generally, you get that protein predicted about right about 2/3 of the time. For that same pose with the best protein-protein docking score, you only get the glue predicted correctly about 1/3 of the time. So that's a bit disappointing. And even more disappointing is if you look at whether you get both the glue and the protein predicted simultaneously correctly, then the best you ever do is 8 out of those 32 with the single best protein-protein docking score. If instead we consider the single prediction that has the best small molecule docking score, here are those answers. And just a quick look will tell you that the situation is even worse. Yeah, sure, we do a better job on the molecular glue, but the protein is less accurately predicted. And then really the best we ever do is 3 out of those 32 where we get the whole system predicted correctly. So a couple things to note from this. The best we ever do at getting the entire system correctly predicted is if we take the single prediction out of 100 with the best protein-protein docking score. If you add a constraint to the larger protein or both proteins, but really just the larger protein, you're going to get the correct answer compared to the crystal structure only 25% of the time. Not a great result with this approach 1 here. If we take a look at the crystal structures and kind of what class they are for the ones where we get everything right, a couple things to note. First of all, there's no cereblon in this. None of those 8 cereblon and glue complexes do we get them both correctly predicted. It's not an entire failure for targeted protein degradation because we can get these decaf 15 structures predicted correctly sometimes. There were 7 of them, and the best we ever do is 3. I think a fair conclusion from this is it's not a great modeling technique. We can get the answer correct sometimes, and we are making it a bit difficult by just taking a single answer as opposed out of that ensemble of 100. So with that, it's not terrible, but we would like to do better, I think is what we can say. So if we go back to kind of our those 3 methods I talked about, those 3 orders of events for piecing together these ternary complexes, just to summarize what I just showed on the previous slide, sometimes we can get both the glue and the protein of interest correctly predicted, but never for cereblon. This is with the approach where we take the 2 proteins first, and then the glue comes in as a second step. What if we try a different order of events? I guess before that, what if we just forget the scoring? Just out of those 100 total predictions, do we ever get cereblon and the glue predicted correctly? Doesn't have to be the top scoring pose, just ever. Out of the 8 known cereblon complexes in this data set, we get 3 of them correctly predicted in 100 shots on goal, 100 predictions. Again, not great. Okay, what if we actually take a look at what actually happens in reality? This would be, and for this, I'm not going to go through the whole 32 test set. I'm just going to really focus on cereblon. What if we take cereblon, we dock the small molecule, that glue, into where it's known to go in cereblon, and we don't have to try too hard here because it really those glues fit really well in the cereblon pocket. So let's just dock that in there. Take that binary complex and add the protein of interest. Like, this is set up for success right here. This is the known order of events. The cereblon and glue fit together wonderfully. How well do we do? Let's make it even easier on ourselves. Let's not consider scoring. Just give me 100 possible answers. How many of them are correct? 2. Out of the 8, we only get 2 correctly predicted. So somehow it's even worse than this approach I just went to. For the sake of completeness, I'll talk about the other one where we do actually dock the glue to the protein of interest. It doesn't super fit well, but you can get poses even though there's not a deep pocket. We only get one correctly placed there, so enough said about that. We tried it, but it's a bit of a dead end. So trying all these different ways to piece together these ternary complexes, none of them seem to work really well. And so, maybe a bit a milquetoast conclusion here, but it's true nonetheless. So cereblon certainly seems to present a challenge from a protein-protein docking perspective. And I will point out that each one of these do involve protein-protein docking. And so, we didn't want to quit there and say like, all right, the best you can get sometimes is 25% accuracy. We do want to do better than that. So we kind of took a deeper dive on what's going on and why is cereblon so challenging from a protein-protein docking perspective. I will say that, just to reiterate what I just said, all three of these order of events, the ways to piece together these ternary complexes, they do all involve protein-protein docking, but it is actually quite a bit different if you actually take a deep look. So for example, in this first order of events and this third one down here, this is what cereblon looks like. I'm showing in space filling just the surface, but it will really illustrate my point. Contrast as to what's going on with the second one here, where we actually add the glue to cereblon first, and then the protein of interest comes in. The protein of interest sees this, whereas in 1 and 3 here, it sees this. And to be more specific, there's a valley here for 1 and 3, and there's a hill or a knob or protuberance, wherever you'd like to call it, in the second approach here. So this is quite a dramatic difference from a protein-protein docking perspective, where you see a valley or a hill. And so that actually got us thinking about kind of reflecting back on work we had done with our bifunctional degraders and how we model those ternary complexes, because there we do a lot of protein-protein docking as well. It actually seems to work pretty well. And so we, better than that 25% accuracy. And so we actually took a look at kind of, this phenomenon here. What is actually being docked as we do that protein-protein docking? When you run our bifunctional model, our bifunctional degrader modeling tools, you have to give it, typically for degradation, you give an E3 ligase with a binder in the pocket. And as we take a look at what those look like, here I'm just showing 2 examples. It's actually this is the input that you give. And there's no crystallographic precedence for essentially most E3 ligases that one would use. This is not cereblon, this is VHL. It's a different E3 ligase, and there's its binder. And here's another E3 ligase with its binder. This E3 ligase is CIEP1. And it's hard to show on the flat slide, but if you can look at these in 3D, it actually turns out that these binders that you give as input, they're certainly not valleys. They're not even really hills. So it's kind of actually intermediate between these 2 extremes. It actually kind of flattens out that potential of protein-protein interaction. So it's neither one of these. So it seems like all 3 of these are inaccurate because that cereblon model we're using is too extreme. And so this actually got us to thinking about a 4th way that we could make these, particularly for cereblon, we could make the inputs here. And that is not to consider that valley or that hill. Instead, it's just to consider part of our molecular glue. We'll call it a glue warhead. And so it's really going to be modeling these molecular glue ternary complexes more or less the same way we do these with bifunctionals in Mo. And that's so really what we're considering in this is these molecular glues are like bifunctionals. So there's really going to be 2 parts, one part interacting with one protein and the second part, excuse me, with the other protein, except there's no linker. And that's really going to be the distinction between glues and heterobifunctional degraders in our approach here. So what does that look like? It means, so here are 2 images I showed on the previous slide with VHL, its binder, CIP1, its binder. What if our input for protein-protein docking with cereblon can be kind of a flattened PPI? So we have part of that molecular glue in the pocket, gives us that flat interface there. Maybe that will give us better results. And of course, we didn't want to just stop with maybe, we actually wanted to try it. And so before I show the results for that, and really, I do also want to show you what the interface looks like. There's 2 challenges in this kind of a bit of a wild idea here is treating glues as linkerless bifunctionals. The first, as I made a point specifically to tell you on one of the early slides, is that when someone talks about a molecular glue, we understand them to be monovalent. And I even said that means the entire glue simultaneously interacts with both proteins. And now I'm kind of telling you, forget about that. We're going to take part of the glue is responsible for binding with one protein and another part with the other protein. So how are we going to do this? How are we going to split a molecular glue into these two different warheads if it's understood to be just a single thing? And so what we did is we actually came up with our, showing you a bit of a preview of our interface here. You provide a library of one or more molecular glues in the form of a Mo database, and we're going to find cut points, ways to split these these glues into two pieces. Bifunctionals we actually split into three pieces: binder 1, binder 2 with the linker. These are linkerless bifunctionals, so quote unquote. So it's just going to be the two binders. And the way we come up with those cut points is we enumerate all single bonds, have to be non-aromatic, so true single bonds. And we can't cut methyl. You can't have just a methyl as a warhead. And we're also not going to carve up a cyclohexyl or cyclopenyl or anything like that. And so really, we're just going to give you a list of single bonds to potentially cut this in half. And then by default, with our automatic partitioning procedure, and I'll talk about it in just a bit more detail in just a minute, we're just going to pick the single bond where if you were to cut it, it will give you two more or less evenly sized warheads. You also have the ability to actually override that and pick a different single bond to cut at. If you want to, for example, this one right here, where my mouse cursor is, would really flatten out that cereblon because this glutaramide type moiety would fit in that cereblon pocket, really kind of seal it shut, give you a flat interface. And then we would say the rest of the glue would be the other warhead, and its primary job is responsible for interacting with the second protein. So a lot of flexibility there. I will say the results I'm going to show today, this adds a bit of an arbitrary factor, and I'm doing benchmarking here, so I did want to be consistent. So all of my results today are really just going to go with this automatic, the single bond, where if it splits the glue into two different warheads, they're going to be as evenly sized as possible. So that kind of answers that second. How do you partition a glue? Well, just try to split it in half the best you can. In a nutshell is how we're going to do that. The second challenge, however, is what are we going to look for the inputs? And to really get specific here, I want to contrast with bifunctional. So when we do our bifunctional or modeling, and if you've heard me talk about this, I know you've heard me say the phrase method 4B. That's our most sophisticated tool, our most accurate tool for modeling ternary complexes with bifunctionals. So let's use that as a basis for comparison. With method 4B for bifunctionals, you need to give me 3 ingredients. Of course, you have to give me bifunctionals. Looking at glues, it's going to be the same thing. Just give me some glues, no problems there. The second, if you're looking to do targeted protein degradation with bifunctionals, you have to give me an E3 ligase with a binder. No problem with bifunctionals. There's a lot of crystal structures for the common E3 ligases, and it's really kind of similar for glues. If you're looking at cereblon, a lot of crystallographic precedent. If you're looking at new E3 ligases, less so, but you could probably do some docking and get that situated. Really, the difference comes on the protein of interest. If you're looking to model ternary complex with a bifunctional, let's say you're trying to degrade a kinase, for example, you know that the binder in the pocket is actually going to be incorporated into your bifunctional, and you know that structure. You probably even have a crystal structure of it. So that's really not a problem. Molecular glues, you're often trying to degrade a protein that doesn't have a deep pocket, so we largely do not have these explicit binary complexes of some sort of binder or glue warhead in a pocket co-crystallize with that second protein. So this is the large difference between bifunctional modeling and molecular glue modeling. And so we kind of came up with a design philosophy as we were looking to build our modeling interface here. Unlike with bifunctionals, you do not have to give me those two binary complexes, E3 ligase and binder, protein of interest and binder. Instead, you give me all the information you have, and the technique, the modeling tool will automatically generate kind of behind the scenes any missing information using reasonable computational approaches. And I really do kind of want to go through what that looks like, because it actually makes quite a bit of difference as far as the accuracy of this technique here. So I want to start off with kind of an edge case here. This is something I just said you probably won't generally have, but it's really going to set the stage as we go through different ways to use this tool. And by the way, that's what the tool looks like. Here's the interface. It exists. You can use it today. You just have to ask me for it, and I'm happy to send it to you. So this is what the interface looks like. So let's start with the edge case where you know what of course you know what your two proteins are. You know where those molecular glue warheads are bound to it. Maybe you have, let's say for example, this one over here is in fact cereblon with this molecule co-crystallized in the pocket. So this is all co-crystallized. And let's say your protein of interest shown here in magenta also maybe did a fragment screening campaign, for example. And so you actually know where this moiety also binds to this. So this is very much, this is the exact same input one would use with our bifunctional modeling tool, except as I've mentioned many times, there's no linker here. So how do you actually use this interface if this is the knowledge you have? How do you pass all the information along to the panel? Well, when you're using this panel, you always have to give me the structure of two proteins. Otherwise, there's nothing to do. You, of course, also always have to give me the molecular glue database as well. Now, in this case, this entity here known as binder site, this is really where's the PPI going to form. It's also where's the molecular glue going to dock. But binder site really is protein. We're looking for protein residues. Which part of cereblon over here is going to be at the PPI, and which part of our protein of interest is also going to be at the PPI. In this case, we know it's near that bound small fragment there, so we can just specify it along. It's these pocket atoms, and maybe it's this loop right here we can pass along to the panel. As we're looking to form the Ternary complex, we're really interested in docking these two fragments into the interface there, and we can dock them using a template docking type approach there, knowing that as we form these Ternary complexes, this entity has to match with this up here, and then this part of the molecular glue has to match up there. And so we pass it along with the setting known as binder pose, and you can explicitly give it to the panel. And so you've really given all the information you have about the system to this panel here. Once you have that, the modeling procedure really is the same we do with method 4B for bifunctionals. We do protein-protein docking of these two binary complexes against each other. That gives us how these two proteins might fit together. We also do a pretty quick conformational scan. It's really just a torsion scan at this cut point. So it looks something like this if I'm just superposing my conformations on one entity and letting the other half of the glue spin about. So this is our small conformational ensemble, which gives you some flexibility at that join point right there. And we take these two ensembles together, protein-protein docking, conformational search database, put them together, and that gives you ternary complexes, and those are your answers there, and they get written to a molecular a mode database that you specify down here. Now, let's go to a more realistic example. This would still be kind of a later stage discovery project, and you know the following. Let's say, for the sake of argument, again, you're working with cereblon. We know a lot about that. However, for our protein of interest, the target we're trying to degrade here, we don't actually know how the molecular glue is going to interface with the protein. Let's say we do know where that PPI should form. And in this case, we're just going to say, well, this little moiety down here, it's a beta hairpin. It's a known zinc finger motif that is known to interact with cereblon through homology, structural homology, sequence homology. And so we know that this is where the PPI is going to form, or at least that's what we're going to give to the panel. So basically, the part that's missing is how is the glue going to interact in this area right here near this yellow ribbon right there. So we don't specify it. We leave it as the default value of unknown. We pass the rest of the information along to the panel as we did before. So really, we have to figure out how is this glue warhead going to bind to this protein of interest right here. And the way we're going to do it is we're actually going to do small molecule docking. And we're going to do it in a way where the reconnection point, so right after this amide nitrogen right here, has to be solvent exposed. So we're going to dock that fragment there, and we're going to make sure that the reconnection point to the rest of the glue is sticking out into solvent. However, as you do that, here's one possible example, or possible answer rather. I know it looks like that's buried there. It's just the trick of the 2D slide. It's actually kind of sticking out the backside. So imagine it going into your monitor, but that reconnection point shown with the sphere, it is actually solvent exposed. But this is just one example. This is not a deep traditional pocket here. here, if you look at the docking scores and you use our scoring function, it gives you a score of like -4 or -5, where traditionally with inhibitors, you tend to see like -8, -9, -10. So it fits, it's compatible, but it's not a really tightly held pose, as you can imagine. And so what that means is maybe you don't have a whole lot of confidence in that docking score in the docked pose here. So what do you do about that? Well, we also give you the ability on the panel with this little input section here to create multiple ways, multiple hypotheses for how this molecular glue warhead might bind to that protein of interest. I will say that because we specified this POI binding site, that's the part of the magenta protein that has to interact with cereblon, and it's actually this loop down here, this beta hairpin. We're only going to try to dock this warhead near to that loop right there. So you can see they're all in basically the same area, just different orientations of that molecular glue warhead. And so you can follow the sphere. That's the reconnection point as it explores different areas and kind of this binding groove right here, if you will. So if you turn that input section setting, it can go from 1 to 5. In this case, we only get 4 non-redundant hits there. Now what we can do is we actually have 4 different hypotheses for what this binary complex looks like. And so if we go back to kind of where we started, now our our POI binder pose is unknown, but it's going to be generated behind the scenes automatically with docking. And then we're actually going to try each one of those inputs, and we're going to do multiple simultaneous simulations with those different binary inputs there. And so what we're actually going to do is we're going to repeat the simulation up to 5 times, generate different libraries of ternary complexes, put them all together, try to find commonalities through clustering, and at the end of the day, that's what you get in your output database right here. Now, what another example, I will say that, as you can imagine, running this, like I said, 5 times back to back, it makes it take longer. And so there is the ability to parallelize this job by using our generate batch file. And then you could write a small text summary of what's going on here and run it on a cluster on a cloud, something like that. What if, here's another common situation. Let's say I know all I want to know about, again, let's say cereblon for the sake of argument. My protein of interest, it's brand new. I've got maybe some phenotypic screening or something like that that tells me it's being degraded, but I don't know anything about it beyond maybe like even an alpha fold structure. I have no idea even which face of this protein might interact with cereblon. So I don't know the binder site for my protein of interest, nor its binder pose. And so we have to come up with both of these to give me that binary complex I need to then dock together and run our bifunctional like approach there. So we have to generate both of this information. I'm looking at my clock here. I'm going to skip the details here, but really what we're going to do is we have a way to find sites that the molecular glue warhead might dock. It's kind of based on site finder, as I showed earlier, but also we know that the PPIs between 2 proteins are generally driven by hydrophobicity. So we use our protein patches tool within Mo to find hydrophobic areas, and we really require that those sites be co-located. So it's, as we're looking to dock this glue warhead, it's got to go into a site that works from both a small molecule and a protein perspective. And so we're going to do that. So this automatic procedure, and again, I'm skipping the details, it's going to tell you where the glue warhead might dock. And let's say this is actually the real answer for this exact protein here. I didn't tell it, but it kind of found that same beta hairpin type loop with this algorithm. And then we're going to do the same thing we did before. We're going to small molecule dock the warhead near that yellow binder site there. That'll give us different binder poses. If we turn up the input setting, we'll get multiple hypotheses for the binary complex, and again, collate and cluster to give us ternary complexes there. Another possibility is, this wouldn't really hold true for cereblon, but for things like novel E3 ligases, certainly it would hold true. What if I have some sort of inclination for what the PPI looks like? This might be through something like a hydrogen deuterium exchanger, something along those lines. But I really have no idea how my molecular glues might interact at kind of an atomic level with either one of my proteins. So in this case, I specified both my binder sites. I think it's going to be these orange residues and these yellow residues here, but I don't know how the molecular glue is going to fit with against either one of these proteins here. So we really need to find both binder poses. What we actually do is we actually take those two molecular glue warheads. We don't even know really which warhead goes with which protein. So we really just kind of dock each warhead against those two sites there. And we take the combination that makes the most sense, and that gives us our two binary complexes. So now we're all set. We have two binary complexes, do protein-protein docking, do the conformational, the torsion scan of the glues, try multiple inputs. The same thing I've said a number of times right now. But the point is we've kind of filled in the missing information algorithmically to get you to that point where you can take two binary complexes, do protein-protein docking, and end up with ternary complexes here. Last example is somewhat different. What if this idea sounds too crazy for you? You don't want to do it. You're not interested in splitting your glues. You're a firm believer that these glues are monovalent. No, splitting them, that's not for me. Well, it's really going to be the same interface. You do not have to turn on this partitioning. In fact, it's off by default. And if you do that, then we're actually going to go with the most accurate approach that I've talked about, before I talked about all this. That was that approach 1 that I mentioned, where you're going to take these two apoproteins, protein-protein dock them together to find out potential PPIs, and then that glue comes in as the final step there. And as I mentioned, you can optionally give me binder sites with the same panel. So it really is the same panel. It's just kind of as much information as you have and as much information you're confident in, you can pass it to the panel, and the panel will kind of computationally generate the missing information to give you what it needs, or you can just use it simply, just do some apoprotein-protein docking and then add the glue in to kind of streamline that procedure. Now, I've talked a lot about what this interface looks like. How accurate is this new technique here? And there's a lot of data I generated in looking at this. And it's really, I really wanted to drill down on, should I generate those multiple inputs? As well as I told you, when you do, we cluster the results. Does that clustering add signal, or does it just throw out too much good stuff? So I'm going to summarize that in one slide, and then I'll kind of pull out various lessons on my recommended settings as you use this tool. So what are we looking at? First of all, I will eventually show 4 plots here. They're all going to look, they're going to be structured the same way. There's 4 different bins, 4 different colors here. And these correspond to those examples I just talked about, where, for example, you specify those 2 sites, but nothing about the poses, or you fully specify one protein and not all the other, and then so on and so forth. This green one is where you give all the information you have. You know both binder sites, both binder poses, and that's as good as you can get with this green bars here. The y-axis is going to be our hit rate. That is the same metrics I showed you before. Do we get the protein and the glue within 10 angstrom of the known crystal structure, and then divided by either the total number of ternary complexes, or if we do clustering, how many are in that single largest cluster. And so really what you want to see is a lot of bars, and they should be tall, and that's going to be good for you. And then generally, as you go from left to right, from purple to green, you're adding more information to the simulation, so you're making the job easier. I'll also point out that I'm highlighting these bars in red are the ones that contain cereblon, and this is important to note because we did such a poor job with my earlier approaches on cereblon. But even right now, you can see that there's a lot of red bars here. We are reproducing crystal known structures with cereblon. Clear contrast to what we showed initially. So now I'll turn on the other promised bar graphs here. This is if we do some clustering, and I'll kind of illuminate the differences in just a second. And then what if we try the multiple inputs here? So this is kind of my one snapshot here, and I stared at this thing for hours, went a little crazy. It was like a beautiful mind. Let me share you the results of my insanity on the next few slides here. What lessons can we learn from all these results here? And by the way, I don't think I mentioned, this is that same 32-member test set that we had before. So the first lesson, clustering, should we do it? I would say generally yes. You often do get enrichment, but sometimes your crystal-like poses actually do not fall into the largest cluster, and so your hit rate can actually go down. So, and that's in contrast to our bifunctional modeling tools, where we always cluster. I always recommend clustering for that. Look at that largest cluster. With glues, it's a little less clear-cut. It kind of looks like this. This is a nice example here. You see that sometimes my bars go up, and so I'm getting a better hit rate. That cluster is really enriched for crystal-like poses, but you might notice that there's fewer bars here. So some of them, my hit rate actually falls to 0 because my crystal-like poses in the unclustered results are actually falling into the second largest cluster, the third largest. And here I'm only considering the largest cluster. So a bit of a mixed bag. I would say generally I do tend to cluster, but just know that sometimes your crystal-like poses will fall into not the largest cluster. What about multiple inputs? Generally, I would say it's a good idea. And here's a nice illustration just pulled out from those bar graphs there. Here you can see that our bars are going up. Hit rates are getting better, and we're seeing more bars. So yes, running the multiple inputs generally is a good idea. However, it does take longer to do this. So if you're going to do this, I do recommend parallelizing this. And it also shows less of a benefit if you're giving the information, the simulation more information from the start. So this would be a case where I fully specified one protein, and then that second protein I've indicated its binder site, but not how the glue would interact with it. So here, if you make more inputs, you can see, yeah, it's costing me a little bit. At the high end, I am getting more. So it's a bit of a mixed bag there, but I would say in general, we do recommend more inputs. Maybe you can skip it and not pay the extra computational costs if you're already starting from a pretty mature project and you're already giving the simulation a lot of information. I will say this is a big deal, and it's kind of underappreciated as I've talked to people about this. Ensuring some sort of constraint on both proteins is tremendously beneficial. So for example, here are pulled out from my bar graphs where I'm fully specifying one protein and giving for the other protein, I'm giving no information other than its structure, not giving its binder site or its binder pose. If instead, now I'm saying, okay, now I'm going to give you even the binder site, which face of the protein is going to interact with my fully specified protein, that's night and day. I'm getting so many, my hit rates are better. And for this 32 member test set, I'm getting so many more answers with crystal-like poses there. So it's a huge benefit to be able to constrain at least a little bit both proteins as opposed to just a single one. However, that does require having additional information about your system. And maybe if you don't have that, then you can kind of fake it a little bit by trying that multiple shots on goal, turning up that inputs. So it does give you more bars. The hit rates are about the same. So it's not as good as being able to add a second constraint there, but it's better than nothing. So doing that multiple inputs can be quite beneficial for those low knowledge simulations there. And finally, I mentioned this already, but really let's drive it home. You can model cereblon with this partitioning technique in a way you couldn't with the kind of that initial approach one that I talked about. So here we're looking at scoring. I ran 5 inputs. I'm clustered in that largest cluster. So that's the scoring there. How many of them have crystal-like hits for cereblon? 7 out of the 8, almost all of them. Contrast that to, if you look at scoring, none of them. So obvious benefit for looking at cereblon with this partition, this glue partitioning scheme where we chunk that glue into 2 molecular glue warheads. One last thing here, and then we'll take some questions if we have some time. There's kind of preliminary results, something I'm still actively working on. I'm not quite satisfied, but we are making progress here. And this is scoring. If you've heard me talk about heterobifunctional degraders, a big benefit of using our technique there is being able to predict a priori which degraders should do a good job of degrading, and then which ones don't do such a good job, and which ones are terrible and you shouldn't even make them because you're just going to be wasting your time and you're not going to be able to degrade anything. We would like to do something similar for molecular glues, be able to just give me a library of glues and I'll tell you which ones you should prioritize in the synthesis. Now, being able to develop this is actually a bit hampered by a lack of published data as far as molecular glue libraries and which ones are good degraders and which ones are bad. Nonetheless, we were able to pull together a couple different, like a small handful of known data sets there. And this is just one example. So it's from this recent paper. I know it's a JChem-Inf modeling paper, but it does have experimental results in it nonetheless. This is work from Celgene BMS. And they looked at 29 different molecular glues, and they had DC50 values, Y-mins, Y-max, very similar, how much protein basically was degraded. And so what we were able to do is, of course, bin these experimentally based on whether it degraded less than half of the protein, somewhere between 50 and 90% of the protein, or over 90% of the protein. And so what I'm showing down here, the font color for the numbers, these are just their compound numbers in the paper. Anything in red was shown in that paper to be a bad degrader. Anything yellow was kind of middling to okay. And then anything in green are the glues that you would be interested in as far as a degradation perspective. The background here, the boxes, I have color coded them based on our score, based on, with this this experimental setup here, just how many ternary complexes did we get. And so I just chunked them into the same kind of groups. I took the 9 lowest scoring ones, and those are in this box. The 9 medium scoring ones are in this box, and the 11 with the best predictions are in this green box over here. If this were a perfect prediction, all the reds would be here, all the yellows here, and all the greens here. It's not perfect, but you can see there's obvious enrichment here. And obviously we are doing a good job of for this data set, predicting the effective degraders and really kind of, downplaying the ones that are less effective there. So I'm going to leave up this conclusion slide rather than read through it. It's there for you to digest. And then maybe Antian, if we have a little bit of time, if we have a couple questions, I think we could go ahead and handle them. So I want to thank you all for your attention, and go ahead and take it away, Antian.
Yeah, for sure. So we have one question here saying, what if one of my proteins changes its conformation quite considerably as the ternary complex is formed?
Yeah. That does happen. And the protein I've talked about most today is cereblon, and it's been known to happen. There's been some really lovely cryo-EM work looking at cereblon. It actually has an open conformation and a closed conformation. So that's kind of like an archetypical example of this happening. I will say, as far as the modeling, when we do the protein-protein docking, it's really the structure. Really the backbone atoms you give us, those are preserved throughout. The side chains we repack as we do the protein-protein docking. So if your little side chains can change their orientation as the two proteins come together, that will capture. But if you have this open and closing sort of, loops moving, even if you're trying to degrade a kinase and you have like DFG, DFG in, DFG out, that the simulation itself, with our molecular glue modeling panel, it's not going to capture that. However, you can run multiple simulations. You can try, explore what if I have my cereblon open, what if I have my cereblon closed, and just compare those two simulations. So it's not hopeless. It's just the panel will take exactly the backbone conformation you give and use that throughout.
All right, so we have another one here. So for the molecular glue warheads, can I make them myself and dock them into one or both of the proteins?
Yeah, actually that's so that'll be much like this right here. So this, when I set the stage here, I said, what if we had like a fragment screening campaign and came up with this co-crystal structure? Another way to come up with this, to fully specify the binary complexes, is basically you yourself can manually generate these binary inputs. So rather than kind of this procedure here where, behind the scenes, we let the panel do the docking and come up with poses here, you can actually do it yourself. You can just dock this molecular glue warhead against your protein vectors. You can put it wherever you want. Ideally, you'd put it near where that protein-protein interface is going to form. But now you're not relying on the panel to kind of, like a black box, do the work for you. You can actually look at the warhead. You can look at where it binds. Maybe you're forming some interactions you like. So yes, certainly you can generate those yourselves. And then it's going to be very much like this. The tool is not going to fill in the missing information, because there is no missing information. You have explicitly provided it. And now you get to use your expert eye to say, oh, I like how that fragment is fitting there. I like where the rest of the glue is going to exit off of that. That's what I want. So do it that way. And then you don't have to rely on the black box, like I said.
All right, one last question. I guess this one is very common. So where can I find these tools inside of Mo?
Oh yeah, good point. So I will say, if you're opening up Mo and trying to find this molecular glue menu, you're not going to find it. It is an add-on. We don't charge for it, but it's not part of our core Mo installation, just send me an email, support@chemcomp.com, and I'll right away send you our molecular glue. Well, I'm going to lunch after this, but after that, I'll send you our molecular glue toolkit. It's actually bundled with our bifunctional degrader modeling toolkit. So I'll be happy to send it to you, and we can set up a Zoom and talk in more detail, talk about your system if you'd like, and happy to walk you through how to use that.
All right, so I guess that's all the time we have for today. So thank you very much, Mike, and thank you all for joining us today. So as mentioned, if you have any additional questions or ideas for future webinar topics, please don't hesitate to email us at the email shown on the screen here, support@chemcomp.com. So thank you once again, and goodbye.